Right, thank you very much for the invitation. It's a pleasure to be here. We've already heard a lot of interesting things today, a lot about vision and about echolocation. I want to add another sense to it, and that's the olfaction sense, or the sense of smell. And I would like to venture, even though that could be debated, that there is a difference in the visual world and the olfactory world in that in vision we know much better what we're trying to do than in olfaction. In vision, what we're trying to do, I would venture, is to reconstruct the world around us to gather useful information. In olfaction, we have millions of possible chemicals that we can encounter in our lifetime, and we don't really know what they mean, and we don't really know what it means for chemicals to be similar or dissimilar. It's well known that in the olfactory system of animals, some structurally very different chemicals smell the same, and some very, very similar chemicals that might just be different in their chirality smell different. And that has a reason, I suppose. So we don't really know what we're doing, basically. Now, with that prelude, what I want to talk to you about today is two examples of my work where we're trying to kind of crack that a little open and see what kind of problems we encounter if we look at olfaction instead of vision or hearing, and what we can get as clues from animals, like these two guys here. So I'm going to talk about the Drosophila, and I'm not sure, is this the pointer? Drosophila, the little fruit fly. This is not to scale, okay? And this is a honeybee, okay? So it's two different stories. All right. So to start with Drosophila, we were interested in the question whether, given that there are these millions of possible chemicals that animals can encounter, whether by evolution they would have adapted to the ones that matter. I mean, I think it was a reasonable assumption that this could be. And so we asked the question, if we give a Drosophila loads of different chemicals and we record from all their receptors, would they respond more precisely or more informatively to the things they care about? And that, in the case of Drosophila, would be rotten fruit, something that's fermenting, something that is a lot in vine aromas. So you will see one of our sets of odors is called the vine set. One of the reasons also being that I'm doing this in collaboration with a very applied research institution in Australia, and they're interested in later selling it as some technical application to the wine industry. But the question was, are they more sensitive to those odors than to odors that they would never normally encounter in their lives? So we did these experiments, and I must admit, I'm the computational person, so I didn't do the actual experiments. They were done by Dr. De Bruin in Australia. And what he did is he looked at the nose, if you want, of the fly. So that's the antennae and the pulp. So this is the head of the fly here in the front. You don't see actually the proboscis very well here, but they have one, and they have the pulps on the sides. And there are olfactory receptors in all of these structures. And they have different names. It's almost impossible to read, but it's also not important. But you can see they're all named and all known. Then you can go in and you can insert an electrode into the base of the sensilla where these receptor neurons sit that express these receptors, and then get recordings like this from them. So here you have recordings. These are extracellularly recorded spikes in the neurons sitting in the sensillum. And you can see there are two different ones in there under two different amplitudes here. And this is where the odor is given. And then we just count the number of spikes in the different receptors, and one can identify them by their known profile, how they respond to particular odors and on their position. So it's possible then to get profiles where you have the different receptors here and you have kind of spikes per second in response to an odorant. So you can make this kind of whole map. So you sat there, and I didn't ask him how long it took him. He's very good at it, so I suspect he could do it in a year or so. But it is really still a huge job, and he did this overview of all responses of these receptor types for all these different chemicals. So you have chemicals down here. These are the vine odors, okay? And these are, we call them industrial odors. If you have good eyes, you can see there's all kinds of weird things in there. So for example, we have explosive in there, TNT here, then all kinds of dangerous chemicals that you would have in a chemical plant that could spill out, and you see where this is going. Yeah? So if the flies are able to smell these as well as those, then we could maybe build something out of the fly receptors that's useful in industrial settings. So that was behind why this was also interesting. And so we got these profiles, and here they're shown in like big dark circles mean strong response, and white circles actually mean suppression of the normal baseline activity of the receptor neurons. And you see that it is slightly different. There is more strong responses to the ecologically relevant odors. But there is also some strong responses to these other odors that they would normally not encounter, and their response is pretty much weak or strong to all of them. Yeah? So there is some hint that, oh, maybe there is something going on here where they actually not evolved to just respond to the stuff that's relevant. But to get a better grip on it, that's where I came in. We said, okay, we just need to look at, can they actually distinguish based on this information, can they distinguish all these odorants? Yeah? Can we make a machine learning system that based on this information could distinguish the odors, which would tell us that there's enough information in the sensory perception to do this. And so we went ahead and tried to do that. And it's a very busy slide. But basically what I did here was I looked at all possible combinations of these receptor neurons. And then I grouped it in terms of how many of them I would consider. And that goes along the y-axis here. So you see, I took number of receptors between 1 and 20. So that was the maximum that we had recorded. This shows how many combinations you then can make out of the receptor. So if you take 1 out of 20, there's 20 combinations. If you take 9 out of 20, there's some 167,960 combinations. And I would try them all out, and I used the so-called support vector machine then to classify these different odorants. Left is the vine set, right is the industrial set, and these colored bars are the success rate in recognizing all these odorants. And you see, if I only had one receptor type from the fly, I wouldn't do very good. Yeah? So I get some sort of 10% success rate in recognizing something. But if I go down here, I go up to like some 90% success rate. And the important result for us in asking, have they evolved to do the stuff that's relevant? These look pretty much exactly the same as those. So basically the success rates on the industrial set are the same as on the vine set. So we deduce from that that this could be an example that olfactory systems have not evolved to be specialized to certain needs, but they might have been left generalist to be able to adapt to any kind of environment. That would be one interpretation of this. I mean, I'm hanging myself out of the window a bit there, yeah. But we see that they can do it equally well on stuff they really shouldn't be able to do. Yeah? So that was very interesting. All right. So in terms of summarizing this kind of story, yeah, these Drosophila receptors appear as suitable for ecologically non-relevant odors, the industrial set, as for the ecologically relevant one, yeah, the vine-related ones. Now, so far, this was a theoretical exercise in terms of the scientific question, but they're actually trying to take it to an application. And my collaborators in Australia are developing what they call these cyber-nose, where they're actually taking receptors from an animal and they're changing them genetically to fluoresce and use that as an instrument for an electronic nose. And that's the cyber-nose. Unfortunately, they planned to do this with Drosophila, but unfortunately at the same time when they started with that project, we found, not we found, but it was found that in Drosophila the receptors are actually ion channels, and so I need a membrane to function, and then the whole concept of the cyber-nose fell apart, so they had to go and they do C. elegans now, yeah, the worm. So that's a little side story how it goes in science. Yeah, you think you're on the right track, and then suddenly the animal you want to use is completely useless because they have completely different structure than anyone thought before. You know, it just came out 3 years ago. And we were shortlisted, but we didn't get the award. But I just still put it on there. Right. The second story is about bees. And this is an ongoing story, and I can only show you very preliminary results. And where we're looking at there is that in olfaction, bees have to solve, and animals in general, have to solve a very complicated problem. The first element of that complicated problem is that the typical odors we perceive are not pure chemicals. Yeah, so what we did with the Drosophila, we gave it pure chemicals and looked at all of that, and it was complicated enough. But in the real world, it's more complicated. Each flower has already many, many components in its odorant. So we have mixtures to begin with. That's not enough. If you then have flowers in the field, these mixtures are mixed together from different flowers. So you might have a scene like this, and a bee flies through it and needs to find the mixture within the mixtures, as it were. And the big question is, how can we actually make sense out of this really complex scene? And I would venture, and you can try some introspection on this, that humans to some extent have the same ability to find the mixtures in mixtures. So if you go to the coffee shop in the morning, most people, I think, are able to distinguish that there's coffee and bacon, yeah, if they do bacon, and not some sort of coffee bacon smell. Yeah? Nevertheless, both of these smells have tens of components that we can smell above threshold. Yeah? So we have the mixture of two mixtures. It could have been 20 components or 2 times 10, and somehow we know it's 2 times 10. Now there could be all kinds of reasons why we know that, but this is the question we are after here. Okay. Now, just for illustration, yeah, if you look at a rose odor, they have loads of components in there. There's like different classes of volatiles, and then for each of these classes there would be different ones that are above perception threshold. So we're looking at a very complex mix of things. Okay. Now, another thing that most people don't realize about odors, and I didn't realize about odors before I got stuck into this, is that there's no such thing as like a simple gradient of odor concentration where there's a flower somewhere and that's a very strong smell, and then further away it kind of falls off slowly. The air around us doesn't work that way. What the air around us does, even in a room like this where we don't have any wind, yeah, is that it's all in a turbulent regime. So the Reynolds numbers, if you're a physicist, are in a regime where everything's turbulent. Yeah? It's never like a laminar flow and diffusive regime. So if you wanted in images, this would be the image for that. Yeah? So if you had 2 flowers and they were emitting 2 different odorants, they would mix like this. They wouldn't just nicely mix in a smooth way. And at first, I thought, oh wow, this is getting more complicated as the longer we dig for it. But it turns out that this might actually be an opportunity more than a challenge. And that's the content of this part of the talk. We think that the structure of the odor plume that is very complex might actually help to distinguish between the mixture of mixtures versus a single mixture. And the basic idea of that is that if you come here as a bee and you stick your antenna into this plume, then you would get a little whiff of red, and then a little whiff of blue, and then a little whiff of red again, temporarily distinct. So it wouldn't all come at the same time, but there would be the components of the red odor, might be several ones, but they would all come together, go onto the antenna in one whiff, and then you would get a pause, and then there would be another of the blue, and so on. Sometimes they would coincide, but most of the time you get them separately. And that could actually help distinguishing whether it's one mixture from one, like one big mixture from one source or 2 sources mixing in the air. The reason why we believe this might actually be the case is that we do have now experimental evidence that animals can distinguish very finely between the onset of odor on the antenna. But before I show you that evidence and then come to the conclusions, I thought I'd just demonstrate to you that in a different modality, humans can do the same thing and can use this onset information, the information of defined arrival times of stimuli, to distinguish between conditions. And this is in hearing, and this is a fairly well-known problem that you might have heard of, the so-called cocktail party problem, where people go to cocktail parties and everyone's talking through each other, and you're talking to a communication partner in front of you, and you can still hear them just about, but you can hear them what they're saying, even though it has been shown that the sound volume, like how loud they are, is much less than the noise around. Yeah, so if you focus, you can still kind of pick them out. And there are many elements to that. Yeah, there's some visual cues, and there's the shape of the ear and holding the head in certain directions and so on. But there's also something about the onset of odors being coherent or non-coherent. And that can be demonstrated, and I took this, actually my collaborator found this in the literature, from this paper here, completely unrelated people. And they have a demonstration of that, and I would like to try to play that on the sound, yeah? I hope it's not going to go wrong. And what this is it's a very simple experiment. This image here is a cartoon of this. Basically, each line here represents one frequency of a tone, and you can play them all frequencies together, so they're set on at the same time, and they go for 500 milliseconds, half a second. And that's this sound here. I'll play it in a moment. And the other one, the sound here, is where you leave one of the frequencies out, and you put it in 160 milliseconds delayed. Yeah, so they're a bit later. And let's see whether we can hear the difference and what the difference sound like. Okay. So let's hope it works. So here it comes. So that's the one with the synchronous onset. Oops. And then this one. Okay, who heard the difference? Okay, I see a few hands. Can someone describe what they heard in the second sound that was different from the first?
It was probably a single tone that just came in later, as you described.
Thank you. Well, very well described. Yeah, thank you. So I find the longer I play this, the better I hear it. But I do hear, yeah, there's a single tone coming in late. So you have the almost same sound, yeah, and then there's this kind of sound, yeah. So interesting, yes, we hear two sounds here and we hear one sound there. I mean, it's both mixtures of different frequencies. It's the same frequencies in both cases, and at the end, actually, it should sound the same because physically, these are the two same stimuli. But because one comes in late, our brain interprets that, oh, this must be from a different speaker, yeah, that must be from a different source. Then these must be two tones, and that persists until the end of the stimulus. And we say, no, these are two tones, this is one tone. Even though physically, actually, the stimulus is the same. Just tells us a little bit about that the brain is not trying to reconstruct physics, but using cues like this to make the most likely guess of what's going on. Yeah, and so humans can do that, but notice here 160 milliseconds is a good time to get the effect. Yeah, so we come back to that when we see the insect data. So interesting effect in hearing. Now in the insects we did that with olfaction, and these are the collaborators I worked with at the University of Constance in Germany. Paul Süsker was spearheading this effect, and what they did was they looked at bees and trained them to extend their proboscis. Now this is a very classic learning protocol. Basically the bees are rewarded with sugar water if they respond with proboscis extension towards a particular stimulus that's rewarded, and they should not extend the proboscis towards stimuli that are not rewarded. So bees were trained with a stimulus A, and these are single component odors. In this case it was octanol and nonanol. Nonanol and hexanol. And then obviously you balance it, that you swap the meaning of the two so that you don't have any effects of preferences the bees might have to one or the other odor. So they were trained to one of the odors, A, let's say. It was given 800 milliseconds, they were trained to extend their proboscis. And then they were tested with three different conditions of combinations of the two odors. Either A and B were given together, synchronous at the same time, or A was given first and then B, or B was given first and then A. This was done with different types of delays, but down to 6 milliseconds. And the data down here is from the 6 millisecond delay. To give you an impression what 6 milliseconds within 800 milliseconds are, I did this picture here to scale, which means that you can't really see the delay. Sorry about that, but it's there, yeah? So there's 1 pixel delay here, yeah, on the screen about. So very short delays, and now the question is, do they respond differently to any of these three conditions, or is it all the same to them? So here's the training. In the training, you can see up, it's just, they learn this very quickly. Yeah, this is 3 trials of training, and they start responding. This is the percentage of responding bees, and it goes up to some, 70 or so %. I should say, for those who haven't worked with bees, this doesn't mean that each individual bee only does it unreliably, every 70% time. It's actually an effect where some bees don't learn. Yeah, they're just a bit dumb. So the 70% that respond 100% of the time when they get the odor, and the other 30% they're just hopeless. Okay, so that's the 70% here. Okay, then you take the learning ones and you test them on the new stimuli. Okay, and you see 50% of the ones that learned will respond to the synchronous, as we call it, mixture, yeah, where they both come on at the same time. But many more, and significantly more, respond if one of them is earlier. And interestingly, it's symmetric. Yeah? A first or B first, the rewarded one first or the non-rewarded one first, doesn't matter. When they're not synchronous, they find there's more of the component in there than in the full mixture. Even though also in the full mixture they think is some in there. Right? So it's a gradual thing, but clearly there's an effect where the asynchronicity, yeah, plays a role in that they can see better that there's 2 components. And that one was the rewarded odor, obviously. Right, then we ask ourselves, wow, this is interesting. 6 milliseconds. The spikes in the system are about 2 milliseconds wide. So we're talking about a delay that the brain needs to detect that's about 3 spikes wide. How could that ever do that? And that's where I came into play, and we said, okay, let's do a model of this thing. And we had a very simple hypothesis how this might work. And this simple hypothesis summarized in this little diagram here. And I tried to explain it in a few words because I suspect I might be running out of time soon. The idea was that there could be this little circuit out of local neurons here in green that inhibit each other and are very fast responding neurons. And the idea was that, well, each of the odors would preferably activate one type of receptor neurons, olfactory receptor neurons. And if odor A was activating these, then it would activate this local neuron and would suppress the other local neuron. And this would be some sort of winner-take-all circuit, as we call it, where once this is activated, this one can never escape. So if odor B came in and it was to activate these more than those, it would not activate the local neuron anymore because the other one had one already. Yeah? And the other way around. If B comes first, then this one would go on and suppress this one, and there would be no escape. And at the same time, they would also modify the projection neurons, and these are the neurons that actually report deeper into the brain. Yeah? So these are local neurons that do some processing, and these are the projection neurons projected deeper in the brain. And the pattern would be different if this neuron would be the inhibiting neuron or this one would be the inhibiting neuron. So that was the idea. So there must be some sort of local memory formed by this little circuit, which one was first. That would modify what the response pattern was, and therefore the bees could recognize which one was first, or that there was one first rather than them together. And then we put that all into a fairly complicated model that was a data-driven, bottom-up model of how the bee olfactory system looks like. Here are receptor neurons on the antenna, and then you get into the antennal lobe. This is actually a 3D atlas someone has built of this system, and then I simulated these neurons in the dark gray ones here because we have data for them. And I went through a quite lengthy procedure of making them realistic responses so that I could test that this timing actually worked or didn't work. So I would make sure that these neurons respond like the real neurons respond, and so on. Now, if this was a long scientific talk, now I would go into all the details, but I will spare you all of that, and we'll come to the conclusions that we got from this model, and they were very mixed, to be honest. Yeah. So what we found is that the model could, in principle, reproduce this, and what was encouraging was that our limit of detection was around 5 milliseconds, 6 milliseconds. So that matched the experimental data. But we found that it only captured some of the behavior results, and in particular, this result that it was symmetric, it didn't matter whether the rewarded odor came first or the non-rewarded odor came first, was not fully reproduced. So we still don't fully understand how this works. So the symmetrical behavior result we didn't get. But I should point out that it's maybe not so surprising that we didn't get the full truth yet, because we really don't have the information that we need. Yeah, so for example, we don't actually have any information how mixtures are processed in the system, or even on the receptor level. So there's only hypotheses about these that I implemented, but there's no proof that that's any true. Within that model, half of the parameters are actually unknown, and I made them up. Okay, and yes, and we actually also made a bit of a mistake, and I don't know whether anyone has caught on to it. I've also kind of tried to persuade you of this mistake, but obviously, when the bees respond more to one condition or the other, it doesn't necessarily just reflect that they can recognize the rewarded odor better. It also might reflect that they have been trained for that odor, so therefore the odor is completely differently represented. Yeah, so we have completely ignored that we don't ask them what do you recognize, but we had them trained to recognize something because they like it. Okay, so therefore their response to this particular odor might already have changed during draining, which makes a difference, right? So we have completely ignored that there's some learning and plasticity going on in the system, and have interpreted everything as if it was a sensory perception problem, which is not it's not the case. It's more than a sensory perception problem. It's also a problem of learning. Okay, so with that, I wanted to flash this in front of you. So these are the guys in the fly project I talked about, and these are my collaborators in the bee project, and of course there are some funders involved. Now, to the very end, I also brought something along that actually has nothing to do with my scientific research, but something I built a little bit maybe in the spirit of hacking the senses, to reach out to the general public and demonstrate them how interesting the senses of insects, in this case of bees, are. And it's very similar to some of the stuff we've seen in the other talks. It is a system that's trying to give you the impression how it would be like to look through the compound eye of a bee. Yeah? And this is basically how that approximately looks like. It's better you try it out after the question and answer session. I have it over here. Yeah, there's an Oculus Rift VR system, and there you can look through and see how a bee would see a simulated island with loads of flowers, and you can try to forage with bees' eyes on and see how you get on. So with that, thank you very much for your attention.